So, hi class. Now we are going into second part of chapter 2, which deals with cell division. And what we will study in this second part is why do cell divide, and how they divide in bacteria, how they divide in humans, and what is essential before cell division, that the DNA must replicate. So, but we're not going into the details, but this chapter is very, very important. And so please pay attention. So let me give you some basic concept before I go into this chapter. So the question is, why do cells divide? Now, what I want you to think that, as you know, there are two kinds of cells, like bacterial cells, prokaryote, and human cells, or human cells represents eukaryotes. So if you look, bacterial cells, they divide by asexual means and asexually, and the process is binary fission. What happens in this binary fission is that one bacterial cell divides into two halves, and then these two halves then grow into two dependent bacteria. So the progression of bacterial division is, if this is the y-axis, and this is the x-axis. X-axis, y and x-axis. Then if this is the number of cells and this is time, then bacteria divides with time, and straight line, it's arithmetic in beginning and then it becomes logarithmic. And one of the essential features when that bacteria is dividing is presence of nutrition. For example, if a bacteria that infects or contaminates some milk, it will keep growing until the milk, the nutrition is there in the milk. So nutrition is the limiting factor. In human cells, it is quite interesting if you notice that in human cells, our journey starts as a single cell zygote, which is formed by the fertilization of male and female gamete inside mother's body. And then in 9 months, there's a development of infant. So as compared to bacterial cells, human cells, what happens? The cells divide and then differentiate. What do I mean by differentiate? Is that this zygote will form cells which will develop into brain cells, which is the role is to memory and sense pain or other signals. Kidney cells, it is involved in secretion or removal of waste. Then liver cells, intestinal cells, they are involved in digestion and metabolic process. So what happens? What I'm trying to communicate to you, that human journey starts as a single cell, and then there is cell division, and together with cell division, as compared to bacteria, which is logarithmic, there is differentiation into very specialized organs like brain, heart, intestine, kidney, lungs, liver, and many other, skin. So I would like you to compare skin cells with brain cells. So skin cells keeps dividing because old skin cells are removed and it is replaced by new cells. But brain cells divide very slowly. Brain cells, they divide very slowly because if they divide, that's the reason, because if somebody has stroke, those cells are permanently damaged and difficult to bring them into full-fledged action. If it is a strong stroke. Similarly, heart attack, those heart cells, they don't divide. They have limited capacity to divide. So brain cells, heart cells, liver cells. So if somebody has cirrhosis, it's very hard to regenerate liver cells because they divide very slowly. So on the other hand, skin cells, hair follicles, we grow a couple of inches of hair every weeks or month. So some cells keep dividing, some cells, when we eat and our food is passing through our intestine, small intestine, large intestine, it sheds a lot of skin epithelial cells, and then they are replaced. So those epithelial cells, hair follicle, skin cells, they continuously divide. Whereas brain cells, heart cells, liver cells, they divide very slowly, and as we age, it keeps the capacity to divide becomes lesser and lesser. So growth means cell division essentially, but in bacteria it is a number game. In human cells, given the complexity of our body, cells differentiate into specialized organs. Okay, so but cell division is essence of life. Okay, so let's go to the first slide for this part second. So look at this slide, right? Pay attention, it's very interesting that first there is a hierarchy in our body. Cells, identical cells, they form tissues. Identical tissues forms organs, and then they become a part of a system, and then many systems come together to form body. Let's take an example. For example, neuronal cells, they come together to form nerve tissue, which could be part of brain. Brain is a very specialized organ, and then brain communicates with spinal cord, and then it goes into various kinds of nervous system like voluntary or autonomic nervous system. And it helps it communicates with nervous system, it communicates with circulatory system, digestive system, respiratory system, and support the functioning of our body. Now pay attention to this interesting detail also. In our body, almost 300 billion cells divide every day. We don't know, we don't know. We have many micro injuries, or cells old cells are going off, new cells are coming up, to the extent that almost 200 million cells per minute. So from here you can calculate how many new cells are formed per second, and so it's our body has unique capacity to multiply cells. But again, not all cells multiply at this speed or this pace. Brain cells, as we age, it slows down. Heart, liver, everything. But skin cells, they divide for a longer time. So coming back, how does cells divide? So cell doesn't divide haphazardly. I'm talking about human cells. There is a pathway by which cell divide, and that pathway is generally cyclical, and this is called as cell cycle. And this cell cycle is, it doesn't, again, cycle doesn't happen haphazardly, it happens in phases. So let's say one of the phases could be G1 phase, which is called as growth phase. The second phase could be S phase, that is called as synthesis phase. And here DNA duplicates, because if a cell doesn't duplicate its DNA, the other cell without DNA or chromosome cannot survive, because DNA has all the executive information. So before cell division, where cell division means when a single cell divides into two, the DNA has to duplicate. Technically, DNA duplication is called as replication of DNA. And this is an enzymatic process. This is carried out by the enzyme. If you're a biology major, then you will read more detail about the process of replication in molecular biology class or genetics class, or in both. And then after S phase, there is a phase called as G2 phase, and that is growth phase 2. In the growth phase 2, cells are preparing for division. G2 phase. It's called as growth phase 2. Phase 2. This is growth phase 2. Then the remaining is called as the M phase. M phase means mitotic phase. M phase for mitotic phase. And mitotic phase, in mitotic phase is the phase where what happens that the chromosome which has duplicated in the S phase, chromosomal division occurs in the mitotic phase. And this mitotic phase could lead to formation of finally 2 daughter cells. So it is very essential that when a daughter cells, 2 daughter cells are formed, they get equal nucleus, equal chromosomes, and mitochondria, ribosomes, everything equal. So let's say if this is a cell and this is the nucleus, then mitosis is mainly responsible for the division of chromosome and nucleus. So the division of nucleus is called as karyokinesis. Kinesis. And then after this, the cytoplasm divides. First nucleus divides and then cytoplasm divides. The division of cytoplasm is called as cytokinesis. So division of nucleus is called as karyokinesis, and the division of cytoplasm is called as cytokinesis. Karyokinesis, cytokinesis. Now, I want to go back to the slide and show you that how does DNA replication takes place. So DNA replication takes place when a parent DNA. So what you're seeing here is a parent DNA. That is parent DNA. That is parent DNA. That has to divide. But before division, the DNA has to duplicate because the daughter cells have to receive equal number of chromosome or DNA. So what happens here is that, let's say if this is a parent DNA. DNA is double helix, so I will show for simplicity straight line, and these are connected by hydrogen bond, where A is bonded to T, and G is bonded to C, and A to T, or T to A. So this is connected. This is the parent DNA. Now, when the cell has to divide, the enzyme that will play a major role is called as DNA polymerase. And for the division, will what happened that these parent DNA will separate out, and then a new daughter DNA will be synthesized. New daughter DNA will be synthesized. And this is carried out by the enzyme DNA polymerase. So, so this, I don't want to go into too much detail, because you'll learn in this in more molecular biology or genetics course. So I'll leave this here. And now, so again, before cells divide, the DNA has to duplicate. And that process of duplication is called as DNA replication. And this is carried out by an enzyme called as DNA polymerase. And what is enzyme? I talked to you in part 1, that an enzyme is a process that enhances the rate of reaction. Now, let's go into mitosis and meiosis. But first, I would again, before getting into the details of mitosis and meiosis, I'll give you a basic background. So in human body, there are two kinds of cells. In human body, there are two kinds of cells. One type of cells. One category of cells are called as somatic cells, which are maximum in numbers, and they're responsible for developing organs and tissues, and they are in majority. The second kind of cells are called as germ cells. Germ cells, they are mainly responsible for the generation of gametes. So in males, it will form sperm. This will be formed in testes, and in females, form egg, and they're formed in ovaries. So there are what I was trying to say, that there are two kinds of cells: somatic cells, which mainly are in majority. They form organs and But a minority of cell which is present in reproductive organs, testes in males and ovaries in female, they have cells that can give rise to gametes and sperms and eggs, male and female. So what happens that during fertilization, sperm and eggs, they meet and they lead to the formation of zygote. Right? So one is germ cells responsible for the formation of gametes. Now, somatic cells, which forms organs, they mainly undergo mitosis division, mitosis. And what mitosis is, where the chromosome number remain chromosome number remains same. From parent cells to daughter cells, the number of chromosomes remains same. But in meiosis, which is responsible for the generation of egg and sperm, in meiosis, the chromosome number is reduced. Now reduced to what? It is reduced to half. Why half? Because I mentioned to you in the beginning of the class that our body, each cell in our body, in a somatic cell, they have 46 chromosomes or 23 pairs of chromosomes. Don't get confused here. They're not, all the 46 chromosomes are not different, but they're present as pairs. So when the sperm and eggs are fertilizing, then if the numbers are not reduced from 46 to 23, then our species will be lost because the number of the chromosomes will be doubled, and that's not a good sign. So in meiosis, the sperm has 23 chromosomes, the eggs have 23 chromosomes, and when the fertilization occurs, the total leads to 46 or 23 pair of chromosomes. So 23 comes from father and 23 comes from the mother. But before this happens, meiosis have to happen. If meiosis is not right, then there could be a developmental problem. Okay, so both divisions are important. Mitosis is important for supporting our body, because if you're shedding skin cells, if you're shedding hair follicles, if you're shedding intestinal lining, then it's important to replenish those lost cells through mitosis. And then as we become adult, so there is development of sperm and eggs in males and females, and then after reproduction there's fertilization and it's the beginning of a new generation. And here then species from one generation to the next generation, the chromosome number is maintained. So if meiosis goes wrong, the number of chromosomes from one generation to the next generation is changed, then it will be a problem. So meiosis is very generational inheritance, and mitosis is mainly for maintenance. If a DNA is mutated, mitosis goes wrong, it will lead to the formation of tumor and cancer. In meiosis, if there is a problem with sperm or eggs, then it can lead to the formation of developmental diseases, right? It could be syndrome, Down syndrome, it could be many other possibilities. So both are very important for the survival of human beings. Okay, so now we go into the deep of mitosis. So mitosis doesn't occur randomly, it occurs in phases. So I have a better slide for mitosis. Mitosis occurs in phases. If mitosis is not occurring, the cells are present in interphase, then once there's an urge for the cells to divide, then from interphase they enter prophase and then prometaphase, and from prometaphase to metaphase, anaphase, telophase, and cytokinesis. So let's go over again. If the cells are not dividing or are preparing for division, they are present in interphase. The moment there's a triggering signal for the cells to divide, the cells move from interphase to prophase, prophase to prometaphase, prometaphase to metaphase, anaphase, telophase, and cytokinesis. Now, the way I teach is a little bit different. I teach very objectively because students sometimes they get lost between the phases, they get confused. So I want you to focus on a couple of things when you are studying from interphase to telophase. So I want you to focus on a couple of things. What are the things that I would like you to focus are following. So again, I repeat that moving from interphase to telophase and cytokinesis, you have to focus on. So first is interphase, then prophase, then prometaphase, metaphase, anaphase, and telophase. And after that, cytokinesis. Cytokinesis, as I mentioned to you before, it is the division of cytoplasm. So what are these? So from interphase to telophase, they are mainly involved in the division of nucleus, and that is called as karyokinesis. Cytokinesis is division of cytoplasm. First karyokinesis occurs, then cytokinesis occurs. And what are the phases? The phases are prophase, prometaphase, metaphase, anaphase, and telophase. Now to avoid confusion, I teach very systematically, very objectively, and I want you to focus on a couple of things. One is chromosomal structure. So I would like you to focus on chromosomal structure. Then I would like you to focus on chromosomal movement. Then I would like you to focus on formation of spindle fibers. Okay. Then I would like you to focus on movement of centrioles. Then I would like you to focus on presence of absence of nucleolus and nuclei or nucleus. So these are the criteria that I would like you to focus on each phase. Each phase you have to focus on these salient features. If you focus on this, you will never get confused, whether in genetics class or in molecular biology class, and things will be very clear. And so nucleolus. So let's go back to the. So let's look at interphase. What is the structure of chromosomes? So the chromosomes in this phase, the chromosome, they look very thin. The chromosomes, they look very thin. If you focus, they look like very thin, like a ball of wool. And what you see here within the nucleus, within the nucleus is another organelle called as nucleolus. So in interphase cells, chromosome is thin, and what you see as white boundary, this is called as nuclear envelope. And if you focus, zoom in, you'll see nuclear pore. So all these are present. There is a proper cell, there is a proper nucleus, the nucleus has nucleolus, and chromosomes are very thin. And please focus on this especially. These centrioles, they are present in pair and they are localized at the of the cells. So this is interphase. In interphase, cells may not divide, but if there is any situation where cells need to divide, then cells also prepare for division, because cells have to duplicate many things. They need all the resources. Division is very critical for cells, very crucial. So all the prep goes in the interphase. Now let's transit from interphase to prophase. What you see here. First, the chromosomes that were thin, now they become very thick and shortened. The nucleolus, which was present in interphase, disappears in the prophase. There is no nucleolus. So chromosome becomes thicker, shortened, and focus on these two centrioles. They are moving towards opposite end. So centrioles, if I can draw an analogy, they are moving towards opposite pole, so north pole and south pole. And also if you see, there are fibers being formed. These are called as spindle fibers. They are very much proteinaceous in nature, and you'll see what it does. So centrioles, which was localized here in interphase, moves into south pole and north pole. In prophase, begins to move, and there's a formation of, just like sun rays, there's formation of spindle fibers. Now, from prophase, the cells enter into prometaphase. Now, what happens here? The nucleus has almost, begun to disappear. Nucleus begins to disappear. The nucleus, which was present in prophase and present in interphase, begins to disappear in prometaphase. So if you get a question, in which phase the nucleus disappear, it is prometaphase. And what you see here again, the spindle fibers, now they occupy north pole and south pole. And the spindle fiber, they go and connect to the, so focus here, the spindle fibers goes and connect to each chromosome. So let me show you how it connects. So let's say let's say this is a, I'm drawing, let's say this is a piece to which the chromosomes are attached. So this is the chromosome. This information is very important. Chromosomes are attached, and so this is just for you to understand. This is not a perfect diagram, so but I want you to understand. So this, the central piece is called as centromere. And these, what you see like ears of bunny or rabbit, these are called as chromatids. Now, so let's say this is A, this is chromatid A. This is extension of A on the other side of centromere. This is A plus, A prime, A dash. So now, what is the relationship between A and A prime? A and A prime is they're called as sister chromatids. Sister. They will have identical DNA. Their sister. The relationship between A and A plus, A and A prime is they're called as sister chromatid. So centromere. So spindle fiber, they emerge from centriole and they attach to centromere. This is the place where the spindle fibers attach. And what is how it attaches? It attaches through a structure called as, through a structure which is called as kinetochore. So kinetochore. Now, in what context we are talking here? We are talking in the phase pro-metaphase. In pro-metaphase, the nucleus has almost disappeared, and centrioles, they occupy north pole and south pole. Spindle fibers go and attach to the centromere through a structure called as kinetochore, right? So let's go back to that slide. So you see here, the spindle fibers, they go and attach to a knob-like structure called as kinetochore, which you see here. So so chromosomes that were floating in the nucleus, they get attached to spindle fiber. Okay. So let's see what happens in the next stage. So from pro-metaphase, chromosomes, cells, they enter into metaphase. And what happens in metaphase? In metaphase, the chromosomes, they align themselves on a plate. The chromosomes, they align themselves in a plate, and this plate is called as metaphasic plate. You see here. So if you compare to prometaphase, the chromosomes are disorganized. They're attached, but they're disorganized, haphazard. But when you go to metaphase, the chromosomes are arranged in one line, and this is called as metaphasic plate. It is very critical because after this, the chromosomes will divide. The chromosomes will divide. And when they divide, or they break, they move towards opposite poles. So in anaphase, chromosomes split, the chromosomes split and they move towards opposite pole. And they move because these chromosomes are very difficult to break, but this breakage happens because of the power of spindle fibers, and they move towards opposite pole. And if the chromosomal division is successful, the nucleus is ready to divide into two daughter nucleus in the telophase. So what you see here, whatever we lost going from interphase to prophase is lost, that comes back in telophase. So the daughter nuclei, so one nucleus now give rise to two daughter nuclei, in which there is formation of nucleolus. Remember, we lost the nucleolus in the prophase. There's formation of nuclei and nuclear envelope. There is no spindle fibers, right? There is no spindle fiber. Okay, so these, after this nucleus has divided, cells will undergo cytokinesis. So cytokinesis is the division of cytoplasm. So this is in a nutshell about mitosis. So as I mentioned to you, mitosis is mitosis. In mitosis, what are the salient features of mitosis? The salient feature of mitosis is that in mitosis, so first of all, mitosis, what happens in mitosis is that it occurs in somatic cells. Second, chromosomal number is maintained, right? What do I mean by the chromosomal number is maintained? Means if the parent cell has 46 or 23 pairs of chromosome, then the daughter cells, skin cells, will have same 46 or 23 pair of chromosome. There is no change, right? And so these are the main. So, and what is mitosis needed for? This is needed for growth, repair, mitosis. So journey from zygote to an infant in 9 months inside mother's body, it's because of mitosis mainly. Right? Now if we compare mitosis with meiosis, so meiosis occurs in germ cells. And where are germ cells present? They're present in reproductive organs, testes and versus ovaries in male and female, right? And what happens to the chromosome number? In meiosis, the chromosome number is reduced. And what do I mean by that? Is that from 46 or 23 pairs, the chromosomes are reduced to only 23. So mother's egg and father's sperm, they will contain 23 chromosomes each, and when they fuse, during fertilization, they go back to 46. So chromosome number is reduced. And mitosis is mainly needed for maintaining the chromosome number from one generation to the next, one generation to the next. Now, mitosis is not very simple as, meiosis is not very simple as mitosis. Meiosis takes time, and it occurs in two phases. So let's go a little bit into meiosis. Meiosis, which is responsible for reduction of division, it occurs in two phases. First phase is called as meiosis 1, and the second phase is called as meiosis 2. It is the meiosis 1. It is the meiosis 1 that is the reductional division. In meiosis 2 is just like mitosis. So in the chromosome numbers are maintained in meiosis 2. So how does this occur? So again, this also occurs in phases. So let's learn about in brief about the phases of meiosis 1 and 2. So meiosis 1 has phases called as prophase 1, metaphase 1, anaphase 1, and then telophase 1, and cytokinesis 1. So what happens in the reproductive organ, one of the cells will enter into meiosis 1, and then during the process, this will generate two cells. This will generate two cells, and these two cells will have these two cells which are formed out of meiosis 1 will have half the chromosome. So if the parent cell had let's say 46 chromosome, parent cell, the parent cell had 46 chromosome, then the daughter cells will have 23. So 23 and 23. Reduction division, it takes place in meiosis 1. Okay? So one cell gives rise to two cells. Now the question you can think that how does meiosis 2 occurs? So these two cells that are formed in meiosis 1, they enter into meiosis 2. So what is meiosis 2 composed of? Meiosis 2 is composed of one prophase, meiosis 2, this is meiosis 2, prophase 2, second phase is prometaphase 2, third phase is metaphase 2, fourth phase is anaphase 2, fifth phase is telophase 2, and then cytokinesis 2. So in the end, these two cells from meiosis 1 will give rise to 4 cells. Will give rise to 4 cells, 4 identical cells. 4 identical cells. So now, one more difference to add. In mitosis, we get the formation of 2 cells. But in meiosis, we get the formation of 4 cells, right? Which is less than. So mitosis gives rise to 2 cells. Meiosis, because of meiosis 1 and meiosis 2, it gives rise to 4 cells. So just to recapitulate, there are 2 kinds of cells in human bodies. One is called as somatic cells. The second is called as germ cells. Somatic cells multiply by mitosis, which has prophase, prometaphase, metaphase, anaphase, and telophase, and cytokinesis. Give rise to 2 cells with equal number of chromosomes. In meiosis, it occurs in reproductive organs and that give rise to gametes. The chromosome numbers are reduced by half, and meiosis 1 and meiosis 2, 2-phase, it's biphasic. Prophase 1, prometaphase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis 1, and then meiosis 2. So because of that, 4 cells are formed. And another very interesting thing happens in meiosis, and this interesting thing occurs in prophase. This interesting thing occurs in prophase 1. What is that interesting phase? Just a little bit of detail, that in prophase 1, what happens? In prophase 1, what happens there? So let's say in a family, if there are 2 or 3 kids, right? So what happens that they don't resemble to each other. Although the parents are same, the parent chromosome number are same, but the kids, they differ because of a process called as crossing over, which occurs in prophase 1, and that is responsible for the exchange of chromosomal parts, and that give rise to variation in the family. So remember, don't get confused. Meiosis is responsible for, number 1, maintaining the chromosome number from one generation to the next. And second, it also generates variations. And that variation is important, because if we all are same, then there is no identity, but we all are different. So, and that difference comes from variation. And this variation occurs in meiosis in prophase 1 through a process that is called as crossing over, and the genes are exchanged. So let me go back to the slides. So we covered mitosis. So look at this, and then we covered meiosis 1 and meiosis 2. And what is important, that there are some interesting videos that that you must watch on mitosis and meiosis. So I will post those videos on the Blackboard, and that will make things more simpler and more interesting. So with this, I finish with the second part of chapter 2, and we end our chapter 2 here. Okay. Thank you.